Okay, it's fine for me. It sounds so
You had
dumb.
a busy few weeks. No time to study for the ecology test. No time to study for this.
Yeah, we
Hmm.
cannot
All
minimize
right.
some people in this meeting. That's just awesome. Okay, fine. Okay, so
Okay.
transcriptional regulatory systems. We're starting lecture 7, right? Like I thought that we were gonna do
That's
microarrays, but I don't know if it's actually gonna make any questions about the microarrays.
it's also really easy. So it's not the whole like all the stuff you told about microarrays is that there's just two types and blah blah blah. If it's on the test somehow, I can explain it in 5 minutes.
Okay, cool. That's fine. I mean, if so, but I think that if he put something about microarray, it's probably gonna be freaking what's it called multiple choice. It's not gonna be an actual like.
I don't think it, yeah, because it wasn't even material for this test. And considering he didn't even make a short answer question for it,
Yeah,
kind of
exactly.
makes me, yeah. Anyway, so
Yeah,
Black Operon, are you iffy on any parts about that?
I'm not iffy, but I mean, if you want to go through it, I don't mind
I mean,
it. You know what I mean? Like I'm
I'm pretty
fine with.
confident on Lack, so
I mean, I understand it for the most part. You know, like I know that the trans factors are the ones that what's it called? They affect the lag operon, but they're not like exactly in the operon itself. It's
from what I could tell from the like, I looked up other definitions because his wasn't
me
clear.
too.
I think it's like if it's not DNA, it's a trans factor.
Right. I mean,
Yeah.
but like for example, C CRP, I think cap. Yeah, for example, the repressor LAC-I is a trans factor, but it's not exactly in it's in the operon, but it's far
Oh
away from
no,
actually
but
T.
that's capital L. That's the protein.
Right.
So I
That's
think
what
I
I'm
don't
saying.
know. That's actually I was gonna ask him that actually. That's one of my questions for tomorrow. Like, is the lac-I gene a cis factor? Like, I know the protein is, but I don't know about the gene.
You see that if I mean, if so, I don't think it should be that big of a deal. But like, yeah, LAC-I is a repressor, and I know for the most part that LAC-I just binds into the promoter region of the LAC operon in order to reduce the transcription. Right?
Yeah.
It makes a tetramer.
Oh yeah, yeah, I do.
That's it, right? Like that's as much as you gotta
Pretty
know.
much. When hold on. Was that the one that makes the tetramer, or was that for a different molecule? Oh no, that was
Yeah,
for lactate.
LAC-I makes the
It's
tetramer.
at the bottom. Yep. Because I was remembering the dominant and recessive mutation stuff.
Oh, yeah. Yeah, I get you. But yeah, like I is the one that makes the tetramer, and like I think it was dominant. I forgot the difference. Dominant is the one that affects.
Dominant was if the plasmid or the gene starts making a defective protein, and recessive was if it just stops making the protein.
Right. And then so this one would be the LAC-I operon dominant mutation would be the one that would make like not a good enough tetramer, and so it would not actually repress the gene.
Yeah. And I'm asking him about the bindings and all that because I remember he was a stickler about exact bindings. Like, I wonder if we need to know the lac operon binds at -82 +11
Plus
+401
11.
stuff like that.
Oh, those. I mean, yeah, I mean, I could not tell you, dude. But I mean, it's if you ask him, I'm fine because I know that you'll like let me know or something. But
Yeah.
yeah. Yeah.
Then
Now,
so what else is there? That's really it
allolactose
for life.
is a repressor, right? And allolactose is like a it has to bind to something else. It binds to allolactose to kick it off, right?
Hmm. Hmm.
And
I
that's
hope he
about
doesn't
it.
ask us like what type of operon it is, because like depending on how you look at it, it could be two different things. Like you could have negative inducible, or you could have positive inducible if you look at the cat protein.
Hmm. I know that for CAP to be useful, it needs cyclic AMP, right?
Yeah, so you need low glucose.
Low glucose. Yeah. Yeah. So
You know, I feel like we probably have lactose well enough to not worry about this.
yeah, right. Uh, and I know that the cab binds. Well, that's dumb. Honestly, we can just go straight. The gal. How do you feel about the gal?
I
The
mean,
gal.
oh wait, the whole lecture 7 is a lack. I just realized.
Yeah.
All right, let's see. Gal, the only thing that confuses me is having the major and minor repressor. Like, he doesn't say in lecture which one binds where.
Mm-hmm.
I remember he said there's this. He said something interesting. So there, with the two promoters, apparently you always are making a little bit of this stuff,
Mm-hmm.
but when you have galactose, it switches to the other promoter and you make a bunch of it.
Mm-hmm.
So I need to ask him a lot about like I just don't know where the repressors go. I don't know which operon is which.
I got you. I mean, I know
So
that
I don't know.
operon you're talking about or operator.
Operator, my bad. Yeah.
Operator. Okay, yeah, gotcha. I think the operator E is just for PG1 and PG2, and I let me see
I know, but
the.
like, why do you have an operator for promoters?
I don't know.
That's why, like, I need to ask him about the gal operon. I can't figure it out on my own. I'm just asking him.
You see, I know that RNA polymerase binds to PG1 and PG2, and then it starts transcribing from there. But yeah, I don't know.
Yeah, so I don't know. This is what I'm asking him about because we could sit here thinking about it forever. I tried to, but I
Right,
could not figure shit out on it. So I was like, okay.
right.
And the same thing with like the repressor, like the repressosome thing. It says you have gal R and gal S, but then here it only shows gal R. Like, where's the minor repressor?
I mean, maybe Gallus is not that big of a deal. I mean, but once again, if it comes to like answering a question, like the essay, the essay question, yeah, I would probably say both of them. Because just for
Uh,
completion purposes.
Of all of the operons, I feel like this is the one he'd least likely ask a question about because he's only got
Three
two and a
slides.
half slides on it, really.
Oh yeah, two and a half.
Like
Yeah,
the titration thing is hardly even about gal, it's just in general.
right.
So like, I don't know. I'm asking him about the gal operon, so
Mm-hmm.
we can just skip that
Gotcha.
one.
Aravino.
Aura operon,
Mhm.
that's
How do you feel about that?
I'm pretty sure I got most of it now. It's like
Mm-hmm.
I forgot it was the RC dimer binds to I think I1 and O2 in the
Mm-hmm.
absence of a Robinose. Then when a Robinose comes, it switches to I1, I2 and becomes a promoter.
Right.
And then that activates both the PBAD and the PC, I think. And then
Mm-hmm.
it makes a bunch of RC and then negative feedback happens and it shuts down. I think I got it, honestly.
If you have Aravinos, you'll buy an RC. Yeah, Aravinos, I'm fine with. I,
Like,
I know that
not
depending on the gap between O1 and O2, it it's gonna have a repressive or I mean it will repress or not repress the gene. If you have a 10 base pairs, it produces the dimer, and if you have five base pairs, then it won't repress.
the only weird thing I noticed about the oh yeah, it's apparently I remember the now. He said it was a mistake. The O1 should be an I1. That's what it was. I remember he said in lecture there was a mistake there because like the O1 and O2 don't actually touch each other.
O1 should be I1.
Yeah.
Gotcha. Tryptophan operon. Did you do the questions for the for that he posted? I was going to, but like I said, I couldn't because
My
power.
plan was to do them, but I've been taking so long going through all the lectures. I'm probably going to finish going through the lectures and do them as kind of like a confirmation for what I do know and don't know.
Right. You see,
And then I can focus.
I just did them like in my head as I was going through the slides. I was like, oh, okay, this is that, whatever.
See, that's what I'm doing, but then I want to kind of do it again with like without having the slides in front of me and see what I struggle with.
Right. I feel you.
Yeah. So
No,
trip
tryptophan.
operon,
Mm-hmm. I know
I
it.
don't know. I feel pretty comfortable on this one.
Yeah, I mean, it doesn't have a lot either. Like it's tryptophan and operon has five parts, five proteins, and it's each one is to make a different part of the tryptophan, whatever.
At first, I got little tripped up, like I was with the whole two hairpin conformations it could have, but
Yes,
I
I.
understand now. The only thing I'm not sure about is like, so if you look at the cis factors, it says trip leader and attenuator, but I thought the leader and the attenuator were the same thing.
You
Like
see,
the
the
leader,
leader is. I mean, like it says, the leader is always the first one. The attenuator is right after the leader.
I know, but like if that's the case, then what is the attenuator? Because I thought
I guess it's just
the
a random piece of DNA that has something to do with attenuation.
yeah, no,
'Cause
but the leader is the thing that has the two tryptophans in it, right?
Yep.
And that's what the ribosome gets stuck on if there's not enough tryptophan, and so that's what determines attenuation. So I thought the leader was the attenuator, but he has them listed separately.
Yeah. You see, I just kept it like I know that termination stops at the attenuator, and you don't get tryptophan E, which is for something else.
Yeah. It's one of those like little bitty things. Like I understand the entire concept of the operon. It's just that little detail that I
Right.
feel like maybe due to an English barrier.
Right. And the hairpin structure, I know that both hairpin structures will they both terminate the translation of tryptophan, right?
What, one, two, two? No, no. If you form the double hairpin, you terminate. But if you only form the one hairpin between two and three, you don't terminate.
Right. Yeah, that's you see that yeah, is it here where the whole rival switch thing comes in place, whatever, right?
I don't I don't think so because riboswitch is a little molecule or something else interfering with it. But the the hairpin and the tryptophan operon is only because of how quickly or slowly the ribosome moves.
Right.
I remember I asked them exactly that
Mm-hmm. I gotcha.
because I thought it was a riboswitch too, the way the slides looked, but then he was like, no, no.
Okay, so the thing is, when there's no tryptophan, it creates the 2-3 conformation, but in the slides it says that the ribosome stalls. Like, and that
No,
mean
no,
that.
okay.
Mhm,
So if there is no tryptophan, yeah, it makes a 2-3 conformation
mhm,
because it stalls. Like if there is tryptophan, it would normally stall on part one
right,
roundabout. But if you have enough tryptophan, it zooms right past it and it allows the 1-2 loop to form, which allows the 3-4 loop to form, which is the actual terminator.
right.
Yeah. But
So,
if it gets stuck on one, then two and three have time to fuse and you don't terminate.
okay, so the ribosome just stalls at one, but it just keeps going.
Yeah, like it stalls until it gets the tryptophan to go.
Mm.
Because like if you're low on tryptophan, it's gonna stall for longer than if you're swimming in tryptophan.
Oh, gotcha. I gotcha. Yeah.
Yeah. Like I
Okay.
doubt it's very long, but
Yeah, I get it.
yeah.
I feel you. And you, like you said, when it's abundant, the ribosome is flying and it pretty much flies away.
Yeah, and if they're low, it gets stuck.
Yeah, I got you. Okay, that makes sense.
Yeah.
The rival switch, that's just
I don't know what he like. This was just like a afterthought. It seemed
yeah, it was pretty much yeah.
like small molecules can turn off transcription. Yep.
Yeah, I literally looked that up in Google because I have some notes, but like bacterial uses little conformation changes in the mRNA to determine the translation of a gene utilizing another co-repressor. Like what I got from here is that another co-repressor just like comes in, which makes another confirmation, and that just turns off the transcription or the
Yeah,
trans.
pretty much.
So yeah, that's it for that.
All right. Yeah, we're kind of. It's weird. Like most of this stuff is super easy.
Hmm.
It's just I gotta look through it a few times to get it.
Yeah, I got you. Yeah. Oh, yeah. Same for me. Yeah.
Like I've like lecture 11 is still scaring me, but I haven't looked at it since he lectured on it. So I'm thinking once I like write my notes, I'll be a little bit more comfortable.
Oh, yeah. You see, I was literally on it like 30 minutes ago. So after, I'm pretty sure you're gonna get it just like me. I mean, if I did it, you can. But like, after you read it like a couple of times, you get it. You get the most. I'm pretty sure you're gonna have like more deep questions that I honestly don't have because like I think they're. I don't know if he's actually gonna ask that, but
Well, I just like asking the really weird questions in case he tries to get us, because it's possible.
yeah, I feel you. Yeah, I mean, but also like from the last test, he was not really trying to. I mean, except for that one test, those two questions that were not in the slides, and he never said in class.
The ones that we got wrong, yeah.
Yeah, so
Like, what the hell?
I don't
He
know.
never like he talked about two subunits. He never mentioned that they were manually like separated by people. He never mentioned any of that. He never told us it was one subunit. And then there is certainly an argument to be made for RNA being hybrid
I hybrid, yeah.
clinophragments. Like that's
Yeah, and that I mean, honestly, you're smart by asking the weird questions because honestly, he can literally come with one of those weird questions. But yeah,
uh,
yeah,
translation.
yeah. Initiation. I mean, I know the only part that is kind of important is that the time that the mRNA is going to last depends on the three prime untranslated section of it. Why? I don't know,
Yeah,
but yeah.
yeah. Now, without looking at the slide though, do you remember all this? What all the initiation factors do?
By memory, I know initiation factor 3 brings the 30S subunit. Initiation factor 2 brings formal methionine into the P side, and I know the initiation factor binds into the A side, if I'm not mistaken, right?
IF1, yeah, binds to the A site. IF2 brings the informal methionine.
Using
But
GTP, by the way.
wait, what?
It brings F methionine using GTP.
Yeah, and
Yeah,
then but the difference, IF3 just prevents the 50S from binding to the 30S.
right. And I think doesn't that's the only thing that it does. I thought that it was the one that actually brought the
No,
30S.
no, it just blocks 50 from binding to 30.
So what brings the 30S subunit? Which initiation factor brings the 30S subunit to the actual
None.
kind of
Once
garden?
IF2 hydrolyzes the GTP and
Hmm.
all the initiation factors fall off, and then
Hmm.
between the tRNA and the other stuff, the 50S just comes to it.
No, I'm talking about like the actual 30S subunit coming to like the Shine-Delgado sequence. I'm talking about which initiation factor brings the whole 30S subunit of the ribosome to the Shine-Delgado sequence? That's my question.
He didn't talk about that, as far as I could tell.
You see, as far as I know, I thought it was.
Because IF, well, no, IF3 binds like two
Mhm.
steps before the RNA even gets there.
Yeah, that's why I thought it was because from the slide I was like, oh, maybe this is the bitch, you know. But I mean, I'll take it.
Yeah, I'll ask him that. Does IF2 also trigger RNA binding?
Yeah, and that's all to be known. There's an extra slide just saying that some some proteins utilizes the formal group, some proteins cleave that formal group off, and some others just cleave off the whole methionine.
Hold on, I'm writing down the question. I feel like if it was a short answer, it would be fine. Just say it is at this point the mRNA binds, but I don't know.
I mean, just for completion, you know.
Yeah. And then what else was it? So I could not figure this out on like, does he ever say exactly how many amino acyl tRNA synthetases there are, or like how many tRNAs E. coli has? Because like I watched the lecture. I didn't hear him say it, as far as I remember. And
tRNAs?
like, I wonder if we're gonna have to know how many of them there are.
You're talking about like tRNAs that bring a different
Yeah,
amino acid
like I know there's a different tRNA for every amino acid. Oh no, there's a different tRNA for every codon, right?
for every amino acid. Not
Oh,
code. Well, technically, yeah, because it has a different anti-codon, but it always brings the same amino acid. So I don't know if that's considered two or one. I know that there's
hmm,
at least 20. You know,
jeez. Okay.
that could be another question that you could ask him.
Yeah, actually, I'm looking at it. I already had that written down. How many tRNAs are there? Different amino acid synthetase for each amino acid.
Right,
I'll ask him that.
right. I know that the elongation factors have weird names. It's like EFTU, EFTS, and EFG, right?
Yeah. Honestly,
And
elongation's like the easy. Honestly, all of it's kind of easy.
yeah, for the most part, yeah. You see, this was like the first time that I actually saw like all the initiation factors and the elongation and the termination factors, but they're they're doable.
Like Dr. Lee kind of talked about it, but not much.
Yeah, not really. Well, not
Yeah.
that I remember of it, at least.
There was like one slide on it for the final, and it didn't even end up on the final because there was only like one slide.
Right. Okay, so do you remember exactly what your elongation factor does?
EFTU loads the next tRNA to the A site
Mm-hmm.
and secures it there by hydrolyzing a GTP.
Mm-hmm.
EFTS recycles the two by putting another GTP on it. And
Mm-hmm.
then EFG translocates the whole operation one codon down.
That's pretty crazy. Yeah.
What
And
part? What part's crazy?
how all of this shit works.
Yeah,
Like I would never think
it's
that
kind of whack.
that's actually happening in my body like right now, you know?
In millions and millions of cells. Yeah, it's kind of whack.
Exactly. That's fucking crazy. Now, do you remember what subunit of the rRNA is what moves the amino acid from the P side to the A side?
Yeah, the 23. Yeah, there's
Nice.
only two subunits that do anything.
Right.
I asked him. I remember I asked him, "Do like what does the five subunit do?" And he was just like, "We don't know."
It's just but if we don't know what it does, how do we know that it exists in the first place?
Well, because he
I
was he
mean,
was like, "We don't know what it does, but if it's not there, it doesn't work." So he was just like, "We don't know, but you need it."
oh damn. That's a good field of study then. Shit. Okay,
Same
so
reason. Yeah. All
yes.
right. So what's next?
Let me see tRNA. There was
Slide 18 is all about wobble effect, I think.
yeah, but it in my opinion it's kind of dumb, but it's whatever.
I mean,
Because I
yeah.
mean I've seen this so many times that at this point I'm just like yeah yeah. I know the slide 19 I think is just extra like just for our own knowledge, not really nothing
I
important.
wrote it down in my notebook, but I don't really plan on studying this much because if I see those names, I can just reference the slide.
Oh yeah, and like honestly, I don't think that I like I said, I think that this was just more like a oh yeah, just for your own knowledge, just more amino acids,
Yeah. The only weird thing is both of these use a stop codon for their codon.
right? That's fucking crazy. So I'm pretty sure it's just at the very end. I mean,
And he
obviously.
said when I asked him about that in office hours, he was like, "Oh, we'll come back to it. We have not come back to it."
All right.
Anyways, termination.
Hmm.
Termination. As far as I can tell, it's just RF1 or RF2 bind to the A site, and then RF3 comes and triggers the release.
Yeah, and that's it. I mean, I know that if we don't have a stop codon, then we have this tmRNA that is like it
Yeah,
has a super long anticodon sequence that, and it brings an alanine with it, and at some point it has a stop codon that it actually will stop the transcription.
yeah. Those were kind of interesting, honestly.
Yeah, that was kind of crazy in my opinion. I didn't know about that. Well, then he had that slide for polycistronic mRNA, just knowing that it happens at all times at the same time. Well,
And then polar effects. That I remember in lecture that had me tripped up for a minute. I got it
tell me.
now, but it did have me tripped up.
Go ahead,
I
explain.
mean, do you got it now?
I got it, but I just want to hear your explanation.
Okay. So as far as I can tell, or I looked it up actually, polar effects or polar mutations were any mutation that affects genes downstream of the mutation.
Hmm.
So then you automatically have to include all frame shift mutations because that'll fuck up everything after it.
Yeah. Hmm.
And then he was the nonsense thing. I thought one and two were connected to each other when he was explaining it, so I got confused. But when I
Yeah.
realized that two is a separate point, I was like, oh, it makes sense. So like if a nonsense mutation happens, the ribosome gets pulled off of the thing early, and then the row dependent termination happens, and it shuts down transcription early. So then the later stuff also doesn't get translated.
Oh, so you really don't have a row termination, so everything gets like fucky.
Wait, what?
So since you don't have a since it's row dependent termination, you don't have the row protein, so
Well,
you
no, the row protein's still there to fuck it up. The only thing is normally the ribosome covers up the row binding site while it's being transcribed. But
right.
if you knock the ribosome off from a stop codon, then all of a sudden that section's exposed and the row can form, and then transcription stops, and then you stop all the other translation as well.
Damn, that makes a big old fucking mess. So yeah, that's
And
fine.
then you die.
Yeah. Well, the cell die. Hopefully you don't die. And I know
And then
that there,
antibiotics.
yeah, antibiotics. I thought that antibiotics was the easiest fucking slide because I was like,
You know, the whole like half the next slideshow is about which antibiotics were discovered when and what they each do.
yeah. And honestly, I feel like if there was a question about that, you can
I'm gonna
easily.
look it up. It's not worth learning. Like it's not worth the time and energy to memorize it.
It's really not worth the time or energy to be recognized. Now, and the only thing that I think that he wants us to know is about the cold plasmids, the coalescence, and all that shit.
Okay, yeah, that's worth knowing.
Yeah,
But
exactly.
also it's kind of simple. It's just like, yeah, it kills shit.
Yeah, let me see once again. Gene codes for coalescence, whatever. I, I we'll get to it later,
All
but
right, let's just do the plasmids real quick. So stringents versus relax. So that's just the regulation on how often it's allowed to replicate, right? Like relaxed plasmids are allowed to replicate more.
yeah, exactly because they're relaxed, so they're chill, so you can replicate as much as you want.
Okay. And then fertility plasmids, that's easy. We've seen that like 50 times.
Exactly. The cold plasmids, that's the thing that he wants us to know.
Okay. Yeah. So, from what I can tell, the cold plasmid, the T and the R protein are used explicitly to move around the cytotoxic protein and help it find a target to kill.
All right. Did he
And
say that
then,
once?
yeah, he did. The translocation and the receptor protein are. I need to rewatch it again just to be certain, but
I
I
feel like
think,
they're doing the same because you see in my notes, the only thing I put in that the whole main point that I got from this thing was like the coalescence disrupt the membrane and cuts the DNA. That's and that the plasmid had its own immunity protein, so it doesn't kill the owner cell.
he
You know?
did talk about the T and the R subunits, so we should probably both rewatch that. But
Yeah, I rewatched that myself.
I'm pretty sure T and R just move it around and help find the right place for it to attach to so it can destroy stuff.
Nice. That's pretty destructive.
Mm.
And
And
then
then virulent protein. I mean, I don't think we need to know all these little details about anthrax.
nah, I don't think so. I mean, it'd be cool though, but yeah, no, I don't.
If we do, I'll look it up, and that's a free question though,
Right.
because he really
Virulent.
didn't say anything extra on it in class.
Yeah, and then there's plasmid for plant-associated bacteria. They do the nitrogen fixation. I think that we both know that. And the rest of the
Hmm.
plasmids, the most important ones are probably like the R plasmids, the resistant plasmids. And there's
Yeah,
all the
but he doesn't really say anything about it. He just says, "Oh yeah, they exist."
yeah, and then there's like a million resistant plasmids, whatever, for
Mm.
each. And then there's a whole antibiotic thing that we can literally look up.
Antibiotic, semisynthetic, and chemotherapy. Like that's easy enough. It's just like real, half fake, fake.
Exactly, pretty much. Yeah.
All right. Big history on antibiotics. I don't think he would ask us a date. Like in 1981, what was
Mhm.
discovered?
Nah, this ain't no history class.
Yeah. Killing effect versus static effect. This is the only question I wanted to ask him because talks about this, and then he talks about all these different classes and types of antibiotics. I want to ask him what each type does.
Hmm.
Like I know cell wall synthesis inhibitors are static. I know
Hmm.
ribosome inhibitors are lethal, but like I want to know about the rest of them just in case he asks.
About the killing or the static effect. Yeah, I got you. I mean, he didn't really talk
Yeah,
about it in class. It was just more like, oh yeah, this happens.
true. All
Yeah,
right, broad, narrow, limited. That's pretty easy.
that's a gimme. That I mean,
Yeah,
I'm fine with that. Personally.
I this whole slide is easy, and it makes me really worry that he's just not gonna ask any questions from the slide.
Why?
Like, I don't. There's like it's so easy. Like, why would he ask something so easy?
Yeah, I mean, but also like once again, like 8 out of 10 questions in the last test were like dumb as fuck.
Fair enough. Really, we thought 10 out of 10 were. It's just that, yeah. Oh well.
Yeah. Oh well.
Penicillin. mm,
The
like,
the
yeah.
very last slide. Wait, what were you saying?
I'm just looking at the penicillin stuff. Like,
Uh-huh.
I don't remember the enzyme it inhibits, but if he asks, it's very easy to look up. So,
Just remember, penicillin has fatal lactam rings, which is like a little square,
yeah,
literally.
but like the enzyme it inhibits is glycopeptide transpeptidase. Like, I don't remember that.
Oh, the name of it? No, the the enzyme that inhibits that.
No, that, oh no, beta lactamase inhibits it, but
Yeah, that's what I
I was
was.
talking about the thing that beta lactam inhibits. Never mind. It's not that important.
Oh, what inhibit? Oh, yeah. I mean, I just know that it disrupts cell walls and mostly in gram-positive cells because they are the ones that have a big old cell wall, and gram-negative they
Uh,
don't.
uh. And then
So,
he's got, what were you talking about?
no, I was gonna talk about the chloramphenicol acetyltransferase encoded by CAT. I don't know if that's important or not. I really have no notes on that, so I don't know what happened in the class.
Wait, about the chloramphenicol, what?
Acetyltransferase at the very end, slide 29. It just adds this like acetyl group to the whole chloramphenicol.
And then turns it off, yeah.
Yeah, I don't know if that's important. I have no clue.
So all these classifications, I mean, he doesn't really say anything in the lecture that he doesn't have on the slide. So this is all kind of a look it up if he asks type of thing.
Yeah, kind of.
Like I'm not gonna remember nalidixic acid binds to the subunit A of gyrase, but
Yeah, it's not worth it.
like yeah, you can look it up.
Yeah. Now,
Um,
lecture
yeah.
11, do you want to like go yourself, watch the videos and take your notes before that, or you want to just like look for it right now?
I mean, we can look through it real quick because if you know it and can explain a little bit of it, that would probably help.
I can do a little bit. Yeah. So,
See,
plasmid
first slide that
replication.
counts as replication.
Yep. Well, you know that replication is unidirectional or bidirectional. That you understand that, right?
Yeah. Rolling circle took me a little bit, but I think I get it now.
What's your explanation for it? 'Cause I was literally watching the video for that like an hour ago. Hmm.
Little a little blue protein there, which name I forget. I need to look it up again.
Is that rep
But
C?
rep C, that would be it. Cuts the double-stranded origin, embeds itself in it,
Hmm.
hooks onto one side.
Right.
Then the polymerase goes around the circle,
Yeah.
unwinding DNA as it goes, and then the little rep C attaches to the other side and releases, and it becomes that single-stranded protein. I don't know if the rep C, no, rep C couldn't embed in the thing. It's not DNA. It's a protein. Okay, and
Yep.
then single-strand origin, you just primer, replicate, boom.
Yeah. I mean, that's pretty much it.
Like it took me
Now,
a while because the pictures were kind of tricky to me. I could not wrap my head around these pictures.
oh yeah, me either. I had to watch the video a couple times too. It's fine.
I think I learned from some random
Yeah.
Indian dude
Now,
on YouTube.
control of plasmid replication, regulation, using antisense RNA. You see, I did not understand what the fuck antisense RNA was. Like, for some reason, I completely forgot what it was. It's just pretty much the same RNA, it's just the other.
The
The
other
other.
strand.
Yeah. So I was like, oh, damn, I was struggling just for that. Yeah. Okay, so do you remember this whole RNA2 thing?
No, no, I got to look at this in detail. I remember when I was watching the lecture for it, I was like, wow, all this is so easy. And now that I haven't looked at it, I'm like, wait, what?
Okay, so you see that there's an origin. You see RNA2, I believe, is a protein. Yes. And it binds to the origin. And then when it binds to the origin, it it makes RNase H. Wait, then be cleaved with RNase in order to make it so it can be replicated. Okay, so RNase 2 binds to the origin, which makes RNase H to cleave the DNA, and then and RNase H can also turn RNA2 into a primer, and then you can just start replication from there. I don't know if that was well explained.
I'm sure it would make more sense if I knew anything, but I don't know. I'm gonna have to just watch this. We might be better just to skip over this and look at.
Okay, yeah, but yeah, like after you watch it, you see I can understand it myself, but I don't know how to fucking explain it.
Okay, that works. So we'll just skip to plasmid cure for now. I'll watch over it
Mhm.
and look over it tomorrow. Like look
Just like
over it tonight, and that should
copy
be.
and all that shit. Yeah.
Yeah. All right. Plasmid cure. I get that. Um.
So pretty much it's just that there's loss of plasmids during replication. How do they prevent it? They have these toxin antitoxin systems, and the toxin just kills it kills the bacteria in case that they don't have a plasmid. So
Yeah, because it lasts longer,
yeah,
and
so
then
the plasmid itself has the antitoxin. So if you don't have the antitoxin, it's just gonna kill the bacteria. So you don't
yeah.
you don't produce a lot of bacteria without plasmids because that would be too easy for them to be killed by anything, you know.
Yeah, and then site-specific recombinases. I did not quite get that one.
Yeah, me neither. I was, you see, when you just joined, I was literally about to look at the video of that because I honestly,
Maybe we should just skip this whole lecture and then just look at the others because like it seems like both of us are weak on this subject and need to look at it.
I'm weak on this part except for PCR. PCR, I got it.
PCR is not in this one, is it?
Yeah,
I don't see it.
and plasmid cloning, slide 20, and then from there down all the way to 20 through 30.
I see preventing plasmid cure for 21, 22, plasmid incompatibility for 23.
What the fuck lectures do you have?
I'm on lecture 11.
I'm on lecture 11. You see, preventing plasmid cure is slide 17 for me.
Share your screen.
Give me 1 second. What the fuck? Give me 1 second. Now you see here's the start, plasmid replication, right? Then all of this bullshit. Then on 20, plasmids for cloning.
Wait, 1 sec, Mara. I guess. Do y'all not want it? That's up to y'all if y'all want it or not. I mean, Maya's coming up with me, and she's probably gonna be cooking a lot. Okay, that way. All right. Sorry, my brother called.
So you see, this is the start of the presentation, plasmid replication.
Yeah,
Now you go down, we go through all this
this
stuff.
is all the same. Yeah.
Yeah. Now you just keep going. Just make sure that you don't have like extra slides because maybe you have an old version. I don't know.
Keep going. Still
That's
the same.
yeah. Then the explanation of how the fuck that works. Then prevention of plasmid cure by partitioning. Pretty easy. I can explain that. It's pretty easy. Directed
Wait,
mechanism.
what slide is that? Prevent that one right there, the one you're on.
It's 16. Partition.
Okay, that for me is 21.
What the fuck? Then you have extra slides somewhere. Wait, let's start from the beginning. You see number 1? You following?
Hold on. Now I am. Okay.
That's 2, 3.
Oh, I see the problem.
What's that?
I have the like the rollover from the last slideshow here. So your slide 1 is my slide 6.
Oh, that's why. You see, I don't know. How's that? But I think I deleted it. I believe so. I think probably. I don't know. well, thing is, then it's your 26. Then what I'm talking about. If it's my 20,
Well,
it's your 26.
I don't go to 26. I go to 24. My last one is on shuttle plasma. It's right above you.
Right here. Then it's probably your
Yeah,
next lecture. You see that?
it is my next lecture.
Oh, okay. Cool.
Yeah.
That makes
All
sense.
right, so we'll just look over lecture 11 on our own and come back to that later,
Okay.
I guess.
Now
Wait,
lecture.
hold on. My lecture 12 starts off with mutants and mutations. Where is my stuff on PCR?
Then you probably have to re-download lecture 11.
I just downloaded it too.
You did?
I just downloaded it just now.
Maybe try to
It's not
download.
there.
What?
I just downloaded all of these again.
Try to download 12 again? If not, I can just send it to you. That's no biggie.
It's not in the 12 I just downloaded either. Yeah, I think you just have to send it to me.
Okay, I'll send it to you. Well, actually, I can just do that in one quick thing.
How does that even happen?
What's your email?
jhermo1@lsu.edu.
Okay, well, sorry. Genetics. Should have it now.
Okay, doke. That would be crazy if I walked in the test just totally unprepared for PCR because it wasn't on the slides.
I mean, there are questions for PCR in his
I know. I was just like, where is it?
Yeah, there's 2 questions about it.
All right, well, I will look at that
Yeah,
in
you
a
see
bit.
question 10, how would you design primers for PCR? In class, he only explained that you just do that. You just chemically synthesize specific primers for PCR. But in the slides, there's nothing about it.
Oh, and then in the lecture, in the question, he's asking, how do you do it?
Yeah, exactly. And I'm like, maybe that's something else you want to ask him. Because I honestly,
Yeah, I'm gonna have a late
don't know how to
night.
design primers. And in the lecture, like you can see the whole lecture, he doesn't say how to design. He just like, oh yeah, you just like chemically synthesize them. But I don't think that's a good answer for that question. You know.
I'm gonna have a late night tonight. I gotta look over everything, and then I gotta go through all these questions and at least identify the stuff that I know I can't answer,
Right.
and so I can ask them. Yeah. All right. So
You want to go through PCR or not?
yeah, I guess so.
Because at
Like
that,
I mostly get it. I'm sure.
hmm you see, the only question that I do not get is like, how do you design a primer? I mean,
I did not get your email.
you haven't? The fuck? Sent items. Hold on. How about I send it again?
How'd you spell my last name? Because sometimes I think it's mixed up.
No, I got you because it says Jake J. Ermo. So your
Oh, yeah.
middle name is Jay. What's your middle name? Just
Joseph.
Joseph? Interesting.
Yeah. Tis
Uh,
my dad's name.
mm, interesting.
I gotta do my toenails.
It says that it was just sent, literally.
I'm sure it'll load in a bit.
Just now. Maybe that's why it took a minute. Folder.
Yeah. It's not here yet. All right, we'll come back to that later. And just skip to mutants and mutations.
That's lecture 12 for me. Yep.
Yep.
Okay, Newton.
Is a mutant literally just any organism that gets a mutation?
Yeah, exactly. That's the way I literally think about it.
Like, it seems so obvious, but then with him, sometimes I'm not sure.
Right. Mutation means that a mutant has a change in genetic material. Yeah. Could be DNA or RNA. It's crazy.
Hmm.
Just genotype is is not capital letters, and phenotype is capital letters. Now
Oh, yeah.
there's the whole thing about random mutations and directed change. And then
And
there's two experiments. The one, the Lurian Delbruck experiment, showing that it's not a directed mutation because
and really both of them. So, like, okay, the Luria one showed that it's random,
it's
and
right.
then the Letterberg one showed that it's not directed either.
Exactly.
Okay. Those are pretty
This
easy,
proof that
honestly.
mutations are spontaneous, and that's why, like for example, only two specimens survived. That means that only two specimens were able to mutate. So yeah, it
Yeah,
proves
and
that
then he
it's
went
not
and
directed.
said that the ones in the without antibiotic, you probably also find one or two with immunity.
Pretty much, yeah. I
Yeah,
mean,
I think the experiments I get.
yeah, I mean, it's been completely fucking random, you know?
Yeah.
No.
All right. So then you conclude that it's spontaneous, and then
Hmm.
let's see. Oh wait, the next step is just like how it happens.
Kind of, yeah. Spontaneous mutations, DNA replications error by insertion or deletion, repair errors, modification of nucleotides, deamination. I mean, it's pretty much mostly like, oh yeah, this can happen, you know?
Yeah.
And this can become a mutagen, you know?
Hmm.
Like UV radiation, you know?
Base analogs, alkylating, blah blah blah. I guess we're not gonna have to know too much, just that these exist, because he didn't really explain how they do it, what they do, blah blah blah.
Yeah, exactly. Something
So,
something that I remember he said about base analogs is that they use them to kill cancer cells. So that's how bad they are.
oh yeah.
Yep. Now
And
intercalating.
intercalating agents are, when I asked him, he said they were kind of similar to analogs
Really?
because they get between the base pairs.
Hmm. So yeah, so I'm gonna guess it fucks up the whole transcription or translation part, right?
Maybe I should ask him what the difference is.
Insert between. Yeah.
Like, yeah. Oh, well. Anyway.
Oxidative damage.
Ames test.
Yeah, yeah,
Oh,
yeah.
yeah. Oxidative test. That's easy.
Yeah. The
And then
Ames
the
test.
Ames test is pretty cool. It's easy enough, though.
What was the whole point of the if the mutation?
It's just testing if a substance is a mutagen or not.
Ah, dumb. So I'm gonna guess that if the bacteria can live, then it's not a mutant. It's not a mutagen.
If the bacteria live, it is a mutagen.
It is.
Yeah. So, like, the control are grown about the mutagen, and then they're put on a petri dish with like antibiotic on it, and then
Oh
they
yeah,
usually don't survive.
right, right. You see, in this
Yeah.
example, I remember he was talking about histamine deficient environment, and if bacteria can survive in histamine deficient environment, then yeah, they have a
Oh yeah, yeah. That was what he was talking about. I thought he was talking about antibiotics. Okay, yeah. So if they grow it in the histamine deficient environment,
that means that the
that means they
mutagen.
mutated to become like
Yeah,
self whatever.
yeah, I got you.
I
Now,
can't write.
transition mutations is purine by purine, transversion is purine by pyrimidine, or vice versa. I think we know
That's
that.
gonna trip me up just because I have to think about which ones are purines and pyrimidines each time.
But once again, you can literally look it up on
Yeah,
site,
true.
you know.
I don't know why. I found the dumbest way to remember it a long time ago, and it stuck with me. Pyrimidines have Y's in their name.
Pyrimidines.
Yeah,
Really like
C and T.
CNT, cytosine, and thymine. Oh, nice.
Yeah, I don't know why, but that stuck with me on how to remember it.
Thymine, and by default, uracil. Okay. Okay.
Hmm.
Okay. Okay. That's a nice way to think about it. Thank you. I never thought about
I didn't either. I don't know where I got it from.
it.
Someone or something told me that, and I was like, "Oh my gosh, that makes my life easy."
Yeah, right. I always have to like actively learn which one is which. So now, so that's cool. You ask it, Dr. Lee. I mean, Dr. Lee, fucking Dr. Ding about this whole strand slippage, insertion, and deletion. Do you get it now?
Like, I get it. Like, it's basically just a section of DNA that gets skipped when you're replicating, right?
Yeah, literally
Yeah.
basic as that. That's for deletion, right? Or for insertion you're talking about, or just for both?
Is it for both? Or oh, I see. A deletion is when it happens on the original molecule.
Yeah, on the
It
template,
skips over part of that, and an insertion is when it skips.
on the new synthesized DNA.
I don't know.
The only
This,
thing for
yeah.
me is like, how does that actually happen? Like,
That's
what?
what I was getting at when I was asking him the question. I was like, but I don't, he didn't get it that way, and I was like, oh, whatever.
I feel like I feel like if he doesn't get it that way, it's probably because he's not important. Because like personally, I
Bro,
would love to know like what does RNA polymerase has to do, or
in order to drift past like 20 base pairs.
exactly, like I feel like that's a big fuck up, like it's not supposed to just unhook the whole thing and just come back
Yeah,
on.
especially considering they have that big ass clamp on it that holds the polymerase to the thing. Like, how do you skip that much?
I don't know. Yeah, that's fucking crazy. And I mean, if he doesn't want to explain it, then that's
Like, it's cool, but he didn't explain it.
I
And I
mean,
guess considering how little he explained it, it's probably not that important.
yeah, he probably just wants us to know that that's how it happens. I mean, personally, I didn't
He's,
know that that's how it happened, but yeah, it
I think he
happened.
spent more time on it trying to explain it to me than he did in the actual lecture, so I doubt it's
Oh
that
yeah,
important.
yeah, hell yeah.
Yeah, he was like in and out with that on the actual lecture.
Yep.
This whole slideshow so far, I honestly don't think I'm gonna want to rewatch it. I don't even think it's worth time.
It's really not worth it. Mutations and
Like,
mutagens is pretty easy so far.
all right.
Except for the math part. That part I was like, what the fuck are you talking about?
And
Until
then
the
he put the example up, and I was like, oh
yeah,
my
I
gosh.
got it until the example. Yeah, intermolecular recombination. You
That
remember
one's kind of neat to me. I
really?
don't know how it happens either. I don't. Did he explain how that happens?
Not really. He
It's
was
just
just like,
like a
oh yeah,
loop gets
it
cut
just
out.
recombinate. I mean, technically that's how recombination works. Like two strands just like crisscross each other and then they exchange.
I know, but like it
In
just
this
creates a
case,
random plasmid out of nowhere. Like
yeah,
it's just
exactly.
weird.
In this case, it's the own thing. It just creates a loop out of nowhere and deletes the whole sequence. It's like, oh, okay, bye. But
Yeah.
it's okay. It's
Intramolecular recombination, I don't totally get yet.
DNA replication, recombination between sister chromosomes. If I'm honest with you, I don't really have notes on that, so
I'm gonna ask him about this one, see if we need to know, because I don't follow it at all.
yeah. You see, the only thing I get is like, I mean, but also he uses the same letter, so that's not really helpful for like, because
Exactly. Yeah.
in the Y-shaped thingy, the BCD, they're recombination between sister chromosomes, but the duplication of
I don't know.
direct repeats, like, yes,
I'm gonna just ask him about it because it's one of those things that's just not worth wasting your time trying to figure out.
exactly,
But it's good to ask him.
yeah, exactly. Now, I think we know this. Say a single base mutation, silent mutations, they just change,
Yeah,
not really the amino acids.
I think that's fair to say.
Yeah, missense mutations change the amino
I think
acids,
it's
and
all.
nonsense creates stuff. That's
I think it's also fair to say we know frame shifts after all this
frame
time.
shift, insertion, deletions. We all know that. Let's see what else. Bacterial phenotype, oxotrophic
Okay.
mutants. I mean,
So oxytrophic is just like the wild type, but it's limited in some way.
exactly, pretty much. That I mean, yeah, they just require in a specific environment, or they like you see the lac minus. It cannot use sugar. It cannot use
Yeah,
glucose as sugar. I mean, it's just a mutant that cannot use a certain thing.
yeah. Resistant, that's explanatory.
Yeah,
I
hypersensitive,
swear to fuck, after all of that explaining, I still don't know the difference between these two.
hypersensitive and conditional.
It's
uh,
like if it's temperature sensitive, it's hypersensitive to temperature, man.
Yeah. You see, in class, he explained it only with temperature. So let's just keep. I might just keep it tied to temperature. You know.
I swear to the shit, if he asks a question about this, explain the difference, I'm going to blow a gasket.
Yeah, because he actually spent he spent a lot of time explaining that. So you
But he
spent like 10
didn't
minutes on
say
that.
anything about it. Like he said, he just kept repeating what the slide said.
Exactly. Yeah. I mean, I was there. Shit, I heard all that. I was like, I don't know if this is gonna help.
This they didn't help. That whole 10 minutes, I was like, what?
I mean, honestly, you could. I mean, are you? Well, you're going to office hours and like in person,
I'm not gonna ask. No, online. I'm not asking him again.
right? It's okay. Let's just wish it's not gonna be on the test or a question about it.
At the very least, it's not gonna be a short answer question.
Oh yeah, it's not. So if it's a
Then-
multiple choice, we can finesse it.
and we'll just guess if it says radiation, it's hypersensitive.
Right, exactly. That's what I'm saying. If it's oxygen and radiation, it's hypersensitive. If it's temperature, it's conditional.
Yeah, I don't know. I might watch that part of the lecture again just to see if maybe I missed it, but I swear they're the same thing.
Nah,
And then
I,
when he did finally say, "Yeah, I guess there is some overlap," I was like, "Okay."
I mean, I
I
don't
don't know. I feel
know.
like it's just not going to be a question though, because if he conceded that there is some overlap, it's very hard to make a question out of it.
Exactly, because that means that you could technically be right, but
Yeah.
it's whatever. Well, mutation rate, the first two slides. Don't get me wrong. Like, I mean, the first slide, I did not understand what the fuck he was talking about. I didn't know what the fuck
Yeah.
PI was. I know that PI is the probability of event zero where there's no mutations, and then he talked about a lot of math that I honestly was lost.
Wait. What is PI? What about zero?
PI is the probability of event zero. He called it event zero is the probability of a bacteria having no mutations.
So PI is the probability of having no mutations.
Exactly. So he called it probability of event zero.
Hmm.
Yeah.
And then when I is equal to zero, PI is equal to I don't this doesn't even make sense to me.
Yeah, and PI is equals to e to the power of negative m, and m is the average number.
And then what's the exclamation point next to that I even for?
Factorial.
That's what that means?
Yeah.
Oh,
You know
never
what that
mind.
is, right?
No, I don't know what a factorial is.
Uh, easy way to think about it. If you have like 10 factorial, it's 1 times 2 times 3 times 4 times 5, all the way to 10.
Oh,
So like if you have
there's
three,
no way he's asking a math question on that.
oh no, he's not. But just for your own like knowledge, like
Now I know.
5 factorial,
That's
is multiplying every number
in
that
that sequence by itself
by itself. Yeah.
or
So
by the
1
next
times
one.
2 times 3 times 4 times 5. That's 5 factorial.
That's crazy.
How is that useful? I don't fucking know. I just know it.
And now I'm never gonna forget it either.
Exactly.
All right. Well, so this whole slide is pretty much useless then.
uh, kind
Like
of.
I don't
I don't
understand what the
see him.
graph is saying either.
I mean, the graph is just saying the number m is the average number of mutations per culture. So I remember he explained it, but honestly, I don't think he's gonna ask about it. So I'm not. I'm just gonna take it as you know. And like, if he put, I mean, if he puts like an example of calculating the mutation rate in the test, I would be like blessed because like there's an example and like it's pretty easy to follow.
I mean, yeah, the 26, 27 area, that's fine. It's just 25 is like, what is that?
Yeah, exactly. I mean, I tried to watch the video again, but I was just as confused. So honestly, I don't know. Maybe you can get something out of it. I personally
Mm.
didn't. You know,
So A is the mutation rate.
A is the
I
mutation.
think is A the mutation rate?
I guess so. Let me
Because
see.
it's number of mutated hits by the
Yeah,
number of cells
yeah.
per
A
two.
is the mutation rate. Yeah.
Okay. And M is the other event number of events. Okay.
Average the number of events per target. Yeah. So number of mutations per colony. Let's call it.
Yeah, I don't know. It's probably not that bad.
Yeah,
I'll
sure.
ask him about slide 25, maybe, but
He
might
might
not
give you
even.
an extra explanation of something that might not even be in the test. So like, it's really up to you.
It's probably not worth it.
I mean, just for the sake of you actually knowing what's up in that slide,
I know. It's just I don't know if I care enough at this point.
Right. Okay. So strategies for finding a mutant. Screen. I get that it's just by the phenotype under in different mediums using one plate to see which bacteria will grow in the selective medium. So kind of like the test that Lendenberg did.
This was another bullshit one to me too. Screening and selection. Can you explain to me how that is different? You put screening, you put them on a surface where only the mutant will grow. Selection, you put them on a surface where only the mutant will grow.
uh, uh, shit. I mean, this is something that you might want to like ask him to,
Like,
because you see, they're both doing it with the phenotype. So yeah,
the only thing I can see is
I
the
mean,
screen is comparing a replica with a.
yeah, favorable and unfavorable
All right,
conditions.
so I guess screening is to see exactly which ones are mutants, and selection is just to find the mutants.
Yeah,
All right,
I
I'll
mean,
just count that as a difference.
yeah, I mean that counts as a difference technically.
That's the only logical one I can think of.
Yeah. Now, by catabolic pathways, that catabolic and anabolic pathways, that I didn't really get. I don't know if you were like
Oh,
good on that, because I was literally about like I mean I was on slide 11, lecture 11 when you got in here, so I was about to hit this.
mm,
Because you see, catabolic resistance for toxic selects for mutants failure to grow on a particular carbon or nitrogen oxide source. So I'm gonna guess that anabolic
oh,
pathways it depends on the nutritional supplement and
yeah.
catabolic.
So anabolic pathways, you would be like checking if to see if it's a mutant by growing it on H plus or histidine plus and histidine minus solution.
Right.
And then catabolic pathway isolation would be testing if it can grow on like an arabinose plate versus like a glucose plate. Because
Oh, because
like
they have
because you
different
have to
like.
break that you break down the arabinose or the glucose for energy, but you
Mhm.
use the histidine to make stuff.
Right.
Yeah, that's
Oh, yeah, yeah, yeah.
yeah,
I got you. I got you. I got you. Yeah,
like just catabolism versus anabolism.
yeah.
That's
I
not
got
too
you.
bad.
Yep. Mutants with essential genes. Screen
That
for.
I don't understand. These two I don't quite get.
In the You see, me too. Me neither. So I'm probably gonna watch the video on this too, at least today.
Yeah. But I mean, if this is the only slide on the whole lecture that we have to look at the video for, that's not bad.
Yeah, it's really not that bad.
Yeah.
Epistasis. You remember what epistasis is, right?
Yeah, yeah.
One mutation masks the phenotype. This whole cross-fitting thing, I was very iffy about, but then I kind of got it.
At first I was like, no, this doesn't make sense. And then when he finally brought it all together, I was like, oh, this makes sense.
Okay, cool. Now go ahead, explain.
Okay, so since tryptophan E is missing only the first enzyme in the pathway, it can use the A molecule from both B and D,
Mm-hmm.
and it can finish synthesizing tryptophan just from that alone and can grow from that.
Right.
So, but in addition to that, it also gets D from B,
Mm-hmm.
so it can. Oh, I'm getting lost again. Okay, the point is, as long as it gets the A substrate, it can grow, and both of the other ones are capable of making the A substrate, so it grows on both sides.
Right.
tryptophan D has to have the B protein made in order to finish.
Right.
Now, it's not going to get that from the E protein or the E group because it
Yeah.
can't make the first step. It can't make the second step, but
Right.
it can get it from B, which can make everything but the last step. And then B doesn't grow at all because it has no way to get the final step.
What is the final step for B?
Trip B.
Yeah. Well, what is the final step for trip B?
I don't know. This is one of the last molecules you need to make tryptophan, I guess.
Oh, and trip B. That's why you don't have it, just because it's that. You see, that's why I didn't understand, because I was like, if you don't have trip B, how do, like what, like what? Wouldn't you have?
Okay. Yeah. I think it's like they can't replicate, but when you put them there in the first place, they're okay.
Okay.
I don't know. All I know is the ones who are lacking trip B won't grow, and that's it.
Right. You see, I'm probably
He
just
didn't
gonna.
explain that.
I'm gonna probably like look over the slide 30 and this one again, just for sake.
Yeah, probably. He took so long to explain cross feeding though. Like
Yeah, that's why he took a lot of
he
time.
takes a long time to explain things.
Yeah.
I don't know. I think I can probably explain that again a bit quicker now that I said it once. But yeah,
Yeah, I feel you. I mean, it's fine.
I don't know. Does it make sense though, or did
It
I
makes
explain
sense
it
to me, though.
absolute
Like I
shite?
get it. I get it. I get that you need each enzyme to make the other. I just didn't understand the way he explained it. He started with tryptophan B, and he was saying that tryptophan B could pass each enzyme to tryptophan, the one that was lacking tryptophan E, yada yada yada. And
Yeah,
that's you see the way you explained it. I get it better.
yeah, that was a messy explanation for me.
Hey, it's all good though.
All right.
uh, let's see. That should be the very last. Let me see.
Yeah, we're on the last slide now.
Yeah, and I know that he kind of flew through this one a little bit.
Kinda, yeah. This isn't even that long of a.
Yeah,
It's not very long, and it's kind of simple.
and suppression of mutation, reverting. That's just when you create another mutation that pretty much gets rid of the other mutation, right?
Yeah, and I'm trying to remember. Like, true reversion is kind of self-explanatory. I do wonder though, like, if you have a mutation that just switches one codon to another, and you change the amino acid, I wonder if it counts as a true reversion if you switch it to a different codon that codes for the original amino acid. Like, you have like four codons that code for leucine.
Yeah,
I wonder if a true reversion would count if you switched it between any four of them.
I think it should technically count as it, because if you change
And
it back to another to the same codon that is coding for the same amino
I
acid,
guess,
you really didn't change anything. You just changed the letters, but
yeah. All
technically you
right,
still
so
have the same thing from
like
before. So,
it's a weird technicality, but it's like, what if?
yeah,
Oh, okay. So
so
true reversion based substitution, that's pretty
yeah,
easy.
that's pretty easy.
Frame shift, I gotta.
You see, the only thing that I didn't get from frame shift mutations is like you see that in the middle you add a G, but then in the
Yeah,
final step you got rid of that G. Like
yeah.
it's frame shift, but like where did that G
So
go?
I think it's basically saying you have to have a deletion pretty much exactly where it was inserted
I mean,
for
but
that
then
to
wouldn't
happen.
that just count as a deletion mutation? I mean, well, as an insertion, not a. I mean, it's an insertion that causes a frame shift, so you need a deletion to restore it back,
Yeah,
pretty much.
it reverts to frame shift, right? Frame shift.
Okay, yeah, that I just don't know why he put it as four different things different, but it's
I
technically
know it's like he puts it in three different bolded points, but it's all part of the same thing.
exactly. So that just threw me off for a second, but it's fine.
Uh, and then, all right,
I know that now pseudo is when shit starts to go down. You have like
the first part of pseudo was still kind of easy though, like
yeah,
same codon, same nucleotide, same codon, different nucleotide.
yeah. The only point is like at least you make. I mean, hold on, same codon, same nucleus. Yeah, like at
The
least you
point is,
have
like, I think it's he's like the whole thing he said the whole lecture was like it's better to make something than nothing.
exactly. Yeah, Yeah, that's what I was thinking. Yeah, that's what I wrote down. I mean,
That's
that
like the only thing I wrote down too.
it's better to have an active protein rather than a short useless protein.
Yeah.
Yeah,
And
and
then
that's pretty much for it. Same codon, same nucleotide, different nucleotide. Like it don't matter if you have the same freaking amino acid
Mhm.
or not. As long as you have something, it's fine.
and then intragenic suppressors is then that's where it gets kind of wacky for me.
Yeah, they introgenic different codon in the same gene. So it reversed the mutation of the first mutation, but it doesn't change that codon. It changes another codon. So when you change the first codon, you have, let's say, a protein that is not useful for shit, right? Even though that's kind of not real, but let's think that the first mutant is not useful in because that's just so happened to be the active side of something. But then you have another mutation that changes another codon in that same region that makes it just so happen that it makes that part of the region active again. Like if
Oh
it was
yeah,
binding
I
site,
remember now.
it would be like not good if you have one, but if you change another codon and make another one of the mutants, maybe you make it an active site again.
Oh yeah, that. This is when he was really talking about it being like better to have a 50% active protein than a 0% active protein.
Yeah,
So
that,
I
that,
think the whole point was pseudo doesn't actually fully restore function; it just kind of restores function.
exactly. Yeah,
Okay.
that's why there's 3A and 3B introgenic suppressors that affect protein structure. Like there's some of them that just like
See,
fuck up
this
the
I
whole
need to look at.
structure, but if you add an extra gene, then at least it's a fucky protein, but it kind of does something. You
So
see?
this one I'd have to look at again. So, hmm.
Let me see. Let me see. Let me see. Let me see.
So if it's a structure gene, then all right. So the first step is you get a mutation that fucks it up. Then you get a second mutation on a different codon that unfucks it up. And then if you fix the first mutation, it's going to be fucked up again if it's a structure protein fix.
Yep.
And then if you replace it with the original and it stays unfucked up, it was a protein function mutation.
Yeah. Exactly. That's exactly what it is.
I wish you could use the term fucked up on short answers and tests. It
It
would
would
save
be
so
so
much time.
useful. It would save so much. I swear to God, dude. I can
Like
just
this
like
whole process can be explained in a few sentences if you can say fucked up.
it would be so useful for like ecology or a professor asking 6 words and short answers.
I
Shit.
know it gets fucked up.
It gets fucked up. Shit. Oh my damn.
Mm.
Okay, so intramolecular frameshift mutation suppression that you add an extra. I mean, dude, we know this shit. Come on now.
Oh no,
You know,
wait, this one was. Hold on, I need to look at this one real quick. So, hold on. Oh, this was just like if you have one frame shift
yeah,
and
and
then
that makes a
you
stop
add
codon.
another one.
Yeah, add another one so that you don't have a stop codon, so you have still like a long protein.
Or did he just? I can't even tell what he changed between the second and third column.
You added an A in the glycine. I mean
Oh wait, no,
the
it looks like you delete a C.
ACC. Well, actually, he changed little bit of this shit. ACC.
I see a deleted C personally.
Yeah. That's what it is. That's what it is. That's what it is.
But then I remember in class he didn't mention a deletion. In class he was talking about you could add another one to fix it.
I mean, I'm gonna guess that adding and deleting counts as the same. I mean, maybe not really because if you add another one, then you have a like you remember that if you have two, if you add
Yeah, if
two
you add one,
nucleotides,
you have to add two more to fix the frames.
exactly. And if you add one, if you add another, you just keep fucking it up.
Now I remember he did say after class that like sometimes adding two is better than just adding one because maybe you fix the stop codon, but
Yeah,
I don't know.
but in this case, what he did, I mean, as you can
In this
see,
case, yeah, he just deleted one,
yeah. So I'm gonna guess that
which
deleting
yeah.
and adding counts as the same.
Okay, so it would be a true revertant if you insert and delete the same thing, and it's a pseudo revertant if you insert and delete different things.
Exactly. Yeah,
All right, that's easy enough.
that's easy. Yeah, we got that. Now, intergenic suppressors, informational nonsense. Oh, I remember this. I remember this.
I am struggling to remember this.
Let's see that. Let's just go through it. It comes back. Okay, so
I'm
nonsense
sure.
suppressors when because it's an anti-codon to pair with one of the stop codons. So yeah, like you just have the anti-codon is fucked. So you say mutation on tRNA, so it binds to the stop codon, but it continues the translation instead of stopping. So this is just like you're making a tRNA that is messed up that binds to a stop codon, but it doesn't stop. It just
Oh yeah, these block stopping.
yep, exactly. And then he just has three pictures for each one because we have UAG as a stop codon, UAA, and UGA as stop codons.
Mm. Oh, yeah, okay. So
Oh,
each of these tRNAs is a mutated or messed up version of a normal one,
yeah,
and it's literally just there to stop it from stopping.
yeah, exactly. Pretty much. Instead of stopping, it's not stopping. It's just letting it keep going.
Okay,
Yeah,
that works.
and the next slide, you see, it prevents the termination of translation. This is just an exact. This is just what the anticodon changes to.
Oh, so the ochre turns from AUU to AUG. Wait, no, AUG is a normal codon,
Yeah,
anticodon.
anticodon. I mean, I don't think he's gonna actually wants us to know this shit, but yeah,
This is confusing though, because like he has, like on the ochre suppressor slide, for example, it's the anticodon for UAA already. Like that is a stop codon, but then it shows it getting replaced with G. Oh wait, I think it's just showing that G got replaced with a U. Never mind.
yeah,
I'm looking at it backwards. The arrows are tripping me up. Okay.
yeah. I feel you. There, most of the times the arrows don't. I mean, you they do it different way sometimes. Yeah,
Like he could have just wrote G changed to U, like, and that'd be cool.
you
All
see.
right. So suppressor sup E is blah blah blah.
Where are you?
I don't remember the sup E, F, B, and L stuff. Like I see it's just it's like is that a gene or
Wait, where are you?
14?
14. Oh yeah, okay. So well, tRNA will compete to release its factor so that you can continue. Oh,
Like are those just genes that code for the messed up tRNAs?
nonsense mutation. Uh, hold on. I think this is just an example of things.
Hmm.
Uh, see, I
Like
can't tell you for sure if I'm honest
they're
with you.
written like genes, so I'm just gonna go with that until I watch it. I gotta
Yeah,
watch this part. So far, most of it I don't think I have to come back and watch, but like that part I probably should.
yeah. I wrote down that tRNA will compete with the release factor so that it can continue translation. UAG, so I think 14. Oh, 14 is just like how, the way it's supposed to be, and 15 is just an example of a mutated version of it. Like you see that in 14, the first one is just the normal. In the first square, is just the normal way, and the bottom one is what UAG would do. It would just stop it. You have a
Oh,
trunc.
maybe it's 13 for you then. I'm talking about like the table. I don't know why our stuff's unsynced.
What table are you talking about? What does it say?
Nonsense suppressor tRNAs.
Oh shit, we're talking about different things. That's why I was like, what the fuck? Yeah, okay, keep going.
What slide number is that for you?
13.
Huh. It's 14 for me. Means I got an extra one somewhere.
Um, I don't understand this shit. Well,
I don't
I
know.
don't understand how the fuck we don't have the same slides. But anyway,
Yeah.
what were you
So is the stuff in italics, is that just like the gene that codes for the messed up tRNA that is the suppressor?
I'm pretty sure. I mean, I'm gonna guess it is. Yeah.
Okay, cool. Move on then.
Now the next part, that's what I was talking about.
I see what you mean, yeah. So like normally it would just keep going, or it would stop going.
Yeah, stop at UAA, but then if you make a insertion, you see, then you have a normal insertion, but then in the next one, it says Amber mutant suppressor, de-amber suppressor strain. It instead of stopping, it just keeps going.
It binds there and it just keeps going, yeah.
Yep. And that's, I mean, that it's just a representation of what would it do, you know?
So it's basically just like without UAG or without the tRNA, it would stop early and be a truncated protein. That's it.
Yeah. And if you do have it, you just keep going pretty much. I mean, you, I don't know if your protein would be useful because it probably is supposed to be that large, but yeah,
Yeah.
that's
Well,
fine.
no. The UAG is an early codon, an early stop codon by mistake. So it's actually totally fine.
Oh, okay, I got you.
Yeah.
Oh, gag tag. Oh, yeah, I got you.
Yeah. Okay. So how does translate to termination stop or terminate?
Tandem codons. Gotta remember the suppressor.
This I might need to watch again.
I know that UAA is the most common termination signal. What happened? I wrote
I don't
down
know what
that.
he means by nonsense. Suppressors are usually sick.
No, hold on. What happens if the most of the times it won't bind to UAA? Produces missense mutation protein suppression is not complete. Failed to terminate. Is this nonsense suppressor? Hold on.
I think I need to watch this again. I don't know what anything on this slide means.
On what slide?
My 18, nonsense suppressors are usually sick.
Produces missense mutant proteins. Suppression is not complete.
Yeah,
Terminate.
like it doesn't make much sense to me.
Hold on. What? Mm. Oh, I think what I remember. What I remember from here is that the repressor mutant tRNA, instead of binding to it would bind to a stop codon, right? Now
Wait,
what it
what?
would, you know that the mutant tRNA we've been talking about, they bind into the stop codon, and instead of stopping, they just let it go, keep going.
Right.
And what happens is that same mutant could bind to the actual like UAA, which is the most common termination signal. Oh, and I mean, it won't bind to the. Oh, wait, fuck. Hold on.
Yeah, I'm confused.
Shit. Yeah, just let's just look at that. You know.
Yeah, maybe ask him about it if I can't figure it out.
Yeah.
Yeah. Okay,
I
we'll
remember,
just
dude. I remember in class I understood this shit. Now I don't remember. Fuck. Because most of the times I don't fucking put answers if I understand what's happening.
every so often he says stuff that's like absolutely vital for you to understand the slide with and if you don't have it, you're just like shit.
Yeah.
All right, we'll just skip the slide and watch it. We gotta watch a few things.
Yeah, I gotta watch a few things too. Yeah, promise. Shit. Now frame shift suppressor, one. Then there's an extra base in tRNA, and yeah, it makes a frame shift because your tRNA is physically making a frame
Oh, it
shift.
adds another.
Yeah.
I don't totally understand how that causes a frame. Oh wait, because it means it skips over 4.
Yeah, like instead of having a codon, you have a whatever the fuck you call that. Yeah,
Is
like the
that the
codon.
what
Like
is
is
that the
that
equivalent of though?
hmm?
Like if you have 4 codons, is that the equivalent of like deleting one or
It yeah, pretty mm, Yeah, it counts as deleting one because you're not really adding. You're just taking one of the nucleotides from one codon. So yeah, you would make a nucleotide one of the codons instead of being three would be two, but then you would get the other one right next to it. So yeah, you start to make it's like acting like as a deletion, you know? Yeah.
Or is it like deleting four? Like, does this thing still carry a amino acid? Because if
Yeah,
it
yeah, yeah,
doesn't,
yeah,
it's kind of
exactly.
like deleting four.
Yeah, like you're not deleting four. You're just like you know that when tRNA comes to the mRNA, it binds into the codon with the anticodon.
Yeah.
Instead of binding to three, it would bind to four. So it would bind to the three from the codon and one from the next codon. So it would be like the leading one in the mRNA. You know what I'm saying?
So, all right, it would be the equivalent of removing one from the rest of it.
Yes.
But
Because
you're not. Yeah, yeah. I think I
because
get it.
we're talking about mRNA, right? Like you know that there's a codon and the tRNA binds to the codon because it's
Yeah,
three.
yeah.
If you have
But if
four,
you go over by one, that means you shifted over, which is kind of like you deleted one because you're
yeah, it's
shifting
kind of like
everything
you
over
deleted
one.
one. So the next codon, instead of being C, like for example, in this one you see CCC, the C from
Yeah.
the first nucleotide of the next codon is gonna be gone. So you're gonna use the first nucleotide from the next codon after that. So
Okay.
yeah, you make a big old frame shift, pretty much.
Okay. Protein interaction suppressor.
I know
This
that.
I get, but I don't.
I understand that. Like from the left side, I remember that he explained that if one of the proteins, like this is thinking that they're working in dimers, so two proteins working together, right?
Yeah, and this one was mostly based on like structural mutations, not
Yeah,
functional
exactly.
ones.
Like if one of the proteins doesn't have like the best affinity, like in the left picture, you see that the protein gets like fucked up, fucky, and it
Yeah,
doesn't have the best affinity with the other protein.
yeah,
What the other protein is gonna do is like gonna mutate and it's gonna carry. You have an extra affinity in order to compensate for that lost
yeah. And then he goes, the
interaction.
next slide is, yeah, I get that. And then the next slide is explaining it.
And size compensation. Yeah, pretty much. Yeah, like
Yeah,
if a protein mm, has a mutation, has an extra part, the other protein is just gonna delete a part of it where that extra part is at, so you can have space for
yeah.
it.
Or if it puts a bigger one, you put a smaller one somewhere else so it still fits.
Exactly. Yeah, pretty much.
Okay. All right.
Size compensation, charge compensation.
Overproduction suppression, I understood.
Oh yeah, that was pretty easy. Like if you have half the activity, just make twice as much. Perfect sense.
Yeah, just
Yeah,
gotta eat a ton.
exactly. That makes perfect sense. Let's see, functional suppression.
Functional, I don't totally get.
Is this with the gal? Yeah. ATP. Yeah. E. If an intermediate is
Wasn't it like UDP galactose, if it builds up, is
yeah,
really dangerous?
yeah. If the intermediate is toxic, then the bacteria inactivates the activity of that enzyme producing the intermediate. Yeah. So what it's gonna do is like if you have too much of that intermediate, that bacteria is just gonna stop the enzyme that is producing that intermediate, pretty much.
Okay, so to fix it, you need to mutate the enzyme before it that's creating the intermediate and
Yeah.
mutate that enzyme so that it stops working and stops making the intermediate.
Exactly.
Okay, that's
I
easy.
mean, I don't know if they actually have to like mutate, but they can just like apparently they can just like inactivate the activity of that enzyme.
And that's like some of these things I wasn't sure because like we're talking about mutations for the most part. So I was like, do you have to mutate it or is it just something they can willfully turn off?
I mean, I want to say some mutation, but I don't know.
And like bypass, I'm pretty sure isn't mutations. It's just the
Oh
one
no, yeah,
enzyme working on something it's not supposed to.
yeah. If there's a mutation in RG, you accumulate too much of that, and then yeah, the bacteria just it says that what I wrote down is that the bacteria generates another enzyme that can utilize the intermediate so that you don't create a small. I mean, you don't hold onto
Yeah.
as
Like
much.
it's not going to be a useful molecule you create, but it's not harming you at least.
Exactly. Yeah.
Yeah. This lecture was pretty easy, honestly.
Yeah, the very last lecture was fine. The only thing that, like I said, the only thing that I kind of struggled with was the math part, but that was fine.
Yeah. So
But yeah,
intergenic suppressors are just things that don't happen to the genome.
yeah.
Okay.
That I mean that
Same thought.
so it's intergenomics. So I guess it's yeah. Technically speaking, it shouldn't be.
Because like that's what it seems like. It's like intragenic is like another mutation in the genome
In
that
the
fixes
gene,
the first one.
incorrect that it's not in the gene. Yeah,
Yeah,
that makes
that's
sense.
easy enough. Okay.
Yeah,
So
and I
now
think
I need to look through lecture 11 mostly. I finished looking through 10.